Chapter 10 is bacteria and viruses, and as it says down here on the bottom, this is kind of like our first part in a category that we call microbiology. We're studying things that are very tiny, and so that's what we call it, microbiology. The one thing I want to say is that next Monday, I'm going to hand out a sheet to you for it's a project that you'll be working on. And the project will be about a infectious disease. And you get to pick from a list of infectious diseases, things that you've heard about, you know, like Ebola, malaria, tetanus, you know, different things like that. Each one will have a different one, and then you'll have certain questions that you will have to answer about this disease, like what organism causes this disease? If you're exposed to this disease, how long does it take until you start showing symptoms? We call that the incubation period. How long, you know, does it typically last until you get over it? Or is there any treatment for it? You know, are there any antibiotics or anything like that that you have to give to this person to have them get over it? People from what age group are more likely to get this disease? All kind of questions like that. So I'll give you this sheet that will have, on the back side will be a list that we will pick from. Everybody will have a different one that you're working from. And then on the front side will be this rubric that I'll actually use to grade your assignment. So you will know what I'm grading on. You'll be able to see what things I'm expecting to have in your report. Okay, so microbiology is just a study literally of microbes. And microbes means just organisms that we have to look at, we can only see them through a microscope. We're actually going to be talking about some things here that are too small to be seen with the microscopes that we have here at school. When we talk about bacteria, there are certain characteristics that all bacteria have in common is that they are very small. And because they are so small, it's hard to see them under a microscope. You have to have a good microscope to be able to see them. So, as such, they're kind of hard to study in any detail. Bacteria, from what we have found, it can live just about anywhere. They can live from where it's really cold to where it's really hot, from where there's very little oxygen to where there's lots of oxygen. There's just a lot of places that they can live. One thing they all have in common is they are prokaryotic. So what does that tell us about the cell, if they're prokaryotic? What does it tell us about the cell? Remember, the other kind was eukaryotic, so these are the prokaryotic. So what does it tell us about the cell? They have what kind of a nucleus?
Membrane bound.
The other one. Non-membrane bound nucleus. And what about the organelle? Also non-membrane bound. So they have a nucleus that doesn't have any nuclear membrane around the outside of it. It has it kind of organized in one spot, but not a membrane around it. And they also have, their organelle don't have membranes around them either. Okay, so that's what it means to be prokaryotic. The kingdoms of bacteria that we're going to talk about are Archaebacteria and then Eubacteria. Archaebacteria are, the first part of it is archae, and if you that's kind of like the first part of archaeology. If you're studying archaeology, you're studying what? Just kind of in general. What are you studying? Like if you're studying archaeology, or arche means what? You're going to, go ahead, Landon, what were you going to say?
Is it like prokaryotic or?
No, what does arche mean? This first part, I thought you had that in your notes, don't you, or not? Yes.
It means ancient.
Means ancient, okay? Now, so arche means ancient, so these are archae bacteria. Why do we call these ancient? Why do we call these bacteria ancient?
That's what evolution / animals and stuff came from.
Okay? So this is coming from an evolutionary worldview, a secular worldview, that this was the very first form of life that evolved here on Earth. Archaebacteria. They were the first. And so that's why they put them in a kingdom called Archaebacteria, because they think that this is the life was the first life on Earth, or from this kingdom. Okay, now what do this particular group of bacteria have? And I think this also enters into why they think of them as being very old, is their cell wall doesn't contain things that are commonly found in cell walls. So they haven't evolved this yet. And the chemical or the substance that they don't have in their cell wall is something called peptidoglycan. It's a long word, peptidoglycan. Yeah, so the archaebacteria don't have this in their cell wall. They have a cell wall, but just not this substance in the cell wall. Now, we often refer to them as extremophiles. So where do you think that idea, what comes with that idea? They like to live in extreme environments, like really cold, really hot, places where you wouldn't ordinarily think living things could exist. They can exist under those kinds of conditions. Okay? So we have some of them that are called thermoacidophiles. Thermo means what? From last year. What does thermo mean? Means heat, right? Means heat. So they can live where it is really hot. This picture up here would be like if you were in Yellowstone and they have the different pools of really hot water. Sometimes the coloring in the pool is caused by bacteria that are thriving in this really hot environment. Ordinarily, as hot as that is, it usually kills most bacteria. But there are some of these that survive in this kind of an environment. Okay? Then we have, this has to do with acid. What does acid do to most living things? It's going to injure it or kill it, right? And so, but they have some of them that can live in an acid environment. That kind of can make them harmful because they can survive when we think that we're trying to kill them, but it doesn't kill them. Okay? There's also some bacteria that are methanogens. Methane. They live off of methane. Methane is the active or the main thing that you find in natural gas that people use, to heat their homes, or that's kind of like the typical thing is for heating homes, hot water heaters and stuff like that, is methane. And then we have some of them that we call halophiles. Halo is a group that we have on the periodic table. The second to the last column in our periodic table is group 17, and they're called halogens. That's where that comes from. They can tolerate those harmful chemicals that are in the halogens. In that, like chlorine, for instance, usually will kill most living things, because that's why we chlorinate the water, to make it sanitary for us to drink. And chlorine also bleaches out things like that, but we have some bacteria that can handle that kind of stuff. So we're not necessarily going to kill them by using those chemicals. The other kingdom of bacteria are the eubacteria, and this is a much larger kingdom. These do have a cell wall like the other ones did, but these have peptidoglycan in their cell wall. So the evolutionists picture these as coming after the others. These finally evolved, their cell wall evolved, and as it evolved, it includes this kind of chemical or substance in their cell wall. In the eubacteria, you have some of them that cause disease. Okay? But not all bacteria cause disease. We're going to talk about some of these things in a moment where they don't cause disease. We actually use them to our benefit in some of the foods that we make and eat. A bacteria that causes a disease is called a pathogen. Okay? Like bacteria, any organism, for instance, that causes a disease is called a pathogen, but bacteria can be called, they're an agent for causing a disease. If you're exposed to this, you're going to get a certain disease. So in this list that you're going to be given next Monday, there will be some of them are bacterial diseases that if you're exposed to these, you'll get this disease. Bacteria, though, are also a very important decomposer organisms. So when things die, all of that organism needs to be broken down into raw materials to make these nutrients available to other living things, like the plants, for instance, that need these nutrients, and they will incorporate it into a new plant. So bacteria are good at decomposing things. Now, is in the whole topic of decomposers here on Earth, the living things that do most of the decomposing are either in the kingdom fungi or they're in this particular kingdom, eubacteria. So those are the ones that do most of the decomposing on Earth. Now, what are some uses of bacteria where they're not pathogenic? Well, there are a lot of them because most bacteria don't cause disease. Most of them don't cause disease. They're not pathogenic. Some of them are actually used in making food. And you probably have eaten food with bacteria in it that are of the good kind. There is, for instance, like in raw milk that you get from a cow, there's bacteria in it. If we don't, do anything with that bacteria in there, it will cause the milk to sour. So, to make milk last longer, we will pasteurize. Notice that on a bottle of milk, it always says that it's pasteurized. Well, we add heat to it because the bacteria that's in milk is not one of those thermophiles that we talked about. It is actually sensitive to heat and will cause it, we can kill it all. So we can kill that bacteria that causes the milk to sour. Now, there is some kinds of bacteria in milk that are actually good for us. And so what they will sometimes do is add that bacteria back to the milk after it's been pasteurized. It's killed all the bacteria, and then we put in the good stuff back in. And the kind of bacteria that they put in milk that's often good for it is a lactobacillus. And we'll talk about the bacillus shape in a moment here. But that's the bacteria that's good for you. You know, if you drink milk with that in it, it's good for you. But some of the bacteria are used to make the different flavors of cheese. Some of you each probably have some flavor of cheese you like. Some people don't like cheese at all. I happen to like most kinds of cheese. I'm not into like Limburger and all those really strong flavored cheeses. But some people really like it. I don't particularly. But the bacteria are responsible for taking the milk and converting it into a kind of cheese. If you eat any yogurt at all, it was made using bacteria. Obviously, good kind of bacteria. Do any of you at your house make your own yogurt? Anybody make one? That was a real popular thing for a while. I remember that people would make their own yogurt because they thought it was much healthier than bought yogurt. I'm not sure it really was, but people would make it. So what they would do when they got to the end of their bacteria, their yogurt, they would save a little bit of it, and then take the ingredients that you need for making yogurt, and take a spoonful of this and put it in there and stir it in. What they were stirring in is the bacteria that makes yogurt. And so now this new batch of yogurt is inoculated with this bacteria from my old. And so you just keep passing this bacteria down to each batch that I made. And so you can make your own yogurt. I've never done it, but there were people that were always doing it, just making their own yogurt from bacteria. Okay, another one is vinegar. So vinegar is actually a bacteria acting on like apple cider. There's apple cider vinegar. They have bacteria that makes it become sour, because apple juice is sweet tasting, right? Yeah, but some of the bacteria will convert it into something that's sour tasting, like vinegar has in it. So here's three things already. Any of you like sauerkraut? It's not my thing. Have I eaten it? Yes, because my mom and dad liked sauerkraut. I think the way they would boil like hot dogs. That's where you eat it, with sauerkraut and hot dogs? Yeah, that was just not my kind of thing. I particularly like my hot dogs actually grilled, personally, but but that's what they liked, Have my wife and I ever had sauerkraut and boiled hot dogs? Nope, because neither one of us likes it. So it's one of those things. I think either you like it or you don't like it. Do you remember how old you were when you first had some of it, or you just
like 10?
Oh, so it's not that terribly long ago, huh? Okay. Yeah, some people, and I think all of us have our preferences for tastes. And so some things to us. Now, also another thing that people don't, that have preferences is texture. Like there are certain things that they like the flavor of, but it has a texture that they find very annoying. It will make them gag, if they have that texture, Some things you just don't expect it to be that texture, and it just turns you off. Somebody had a question or comment over here? Okay, now here is another thing that we do in our part of the country here, is making silage. This is food that we're going to feed to particularly cattle and horses, other animals during the winter months when, and this they prefer this actually over, let's say hay that you give them during the winter. Now, some of you will remember when we were at the other campus, when you drive off of Highway 34 and you'd go straight and you'd curve off to the right, and then you'd finally get up close to the school and you'd turn left. Well, when you were going on this angled road, there were 2 fields of corn planted there. But they weren't the kind of corn that they planted to actually grow corn for the kernels. They were actually growing this because they wanted to convert that into silage. So when the thing had grown to full height, while it was still green, they came through with this machine that cuts it off and chops it into tiny little pieces, probably about this long. You know, everything just chopped up, leaves, stem, and all, stalk and all. I mean, if there's a little premature corn cob on there, it got chopped up too. And they haul that, and then they will either put it into long trenches, where they'll just pile it into it, and in winter, they will, with a front-end loader, will pick up this and dump it in the trough for cattle to eat. And they just love it. It's like eating candy to them. They just, they're pushing each other out of the way because they want to have some. The other way that the newer way of handling it so that it will last longer is they'll put it into these plastic, it's like a tube made out of really heavy plastic, like thicker than garbage bags, you know. And they will fill this whole tube up, and the tube will be, probably about this big around, and then they'll seal it on the ends. And inside of it, the bacteria are breaking down this, kind of decomposing it, and that's where all apparently to the cattle, I think it gives some flavors that they wouldn't get if they just ate, just chewed on corn stalk, and what they really like. But in the process, it generates quite a bit of heat. So when they were exposed open on the ground in these trenches, you could go by in winter on a really cold morning and you see all the steam coming up from the silage. I know that we would dig some of that up and give it to the cattle, and if our hands would get cold, we just stick with the gloves on, we just stick it into the silage, and we'd warm our hands up because it was nice and warm inside the silage, because it's a decomposition process, but it generates heat in the process, and the heat keeps the bacteria warm and makes them continue to be active even during the cold winter months. So it's a great food. So that's just using bacteria in a good way to produce food to feed cattle in winter. Now, we also use bacteria for medical supplies. Remember we talked about one of our students is a diabetic, and if you are a diabetic, you need to have what? Constant supply of what? What? Insulin, right? Now, what did we talk about when we were talking about genetics and insulin? Yeah, use gene therapy. We snip out a little piece of gene that has to do with making insulin, and we put it into a bacteria and get the bacteria to make insulin for us, instead of harvesting it from some animal or something like that. So insulin would be one of the things that we get from bacteria. Antibiotics. Some of the antibiotics that we have are either to control bacteria, typically. We've found out, for instance, that certain fungi can produce chemicals that prevent bacteria from multiplying. Some blood proteins we can get from bacteria. Now, the next thing we want to talk about is how we can identify the different bacteria. One of the ways is by shape. Now remember I told you about milk, and there's a lactobacillus was the name of the bacteria, so the bacillus part, I want you to remember that. Okay, one of the shapes is called coccus, and so in the name, often it will have C-O-C-C-I. So if it has C-O-C-C-I in the name, it means that that bacteria is round. It has a round shape. Some of them are individually round, and some of them are actually two of them hooked together, and so they will often put a dye in it in front of it. They call it like Diplococcus, for instance. That means there's two of them connected together. There's another kind that is shaped kind of like this. To me, I always think those are the shape sort of like a burrito that you would get at Chipotle, right? That kind of shape. Obviously not that big, but a little rod shape. So when they talk about rod shape, it doesn't mean a long rod. It's more, it's longer than it is wide. And then we have another kind of bacteria that's in the shape of a spiral, like a corkscrew. And so that is spirillum. So you would, if you find that in the name, that is basically in the name because it's telling us what shape the bacteria is. So when you, if you're, suffering from some illness, and the doctors are trying to find out, what you have, they're going to try to first figure out, is it a bacteria or is it something else? And so they will take swabs or things like that, and then they will grow those, culture those, so that there'll be a lot of them, and then we'll look at them under a microscope and try to find out what shape do they have. That often is a clue as to what diseases it could be. Also eliminates certain diseases as possibilities, because the bacteria are the wrong shape, for instance. The structure of the bacterial cell, remember we said they don't have a nuclear membrane. They do have an area where all the nuclear material is found in one spot, but doesn't have a membrane around the outside. So we would call that a nucleoid region. We can't call it a nucleus because it doesn't have a membrane around it. They also have plasmids. Remember, that was the way that we used to, when we were talking about genetics, that we could use plasmids as a way to bring genetic material into another cell. We'll have that in a moment again. Bacteria have a cell wall. What other living things have cell walls? What other? And there are a lot of them around. What? Plants, right. Plants have cell walls. Okay? Now, this particular cell wall in the eubacteria, remember, is this cell, or the cell membrane, I guess, it had the peptidoglycan. Okay, this is a cell wall in addition to the membrane. They also, some of them have this area around here. They will kind of like secrete this thing around the outside called a capsule. And the purpose of the capsule is to enable the bacteria to survive when environmental conditions don't favor, they're not favorable. Let's say when it's really hot and dry, it'll keep them from drying out and dying. Or if they're exposed to chemicals that would ordinarily kill them, the capsule kind of increases their chances of surviving maybe until conditions are better. So that's what the capsule does out on the outside. So it's not, an actual structure, but, where you can see its parts, it just covers the whole outside with a layer of material. Okay, they also have ribosomes. What do ribosomes do? From a previous chapter? Yes. Yeah, make protein. Protein synthesis. They also need to make protein same way as other cells. Some of them, but not all of them, some of them have flagella. Those are like hairs sticking off that they can whip back and forth like this, and it helps propel them through like the water or a body fluid or something like that. But it helps them to move along. But not all bacteria have them, though. So let's start talking about some kinds of bacteria. First one we're going to talk about is cyanobacteria. Does anybody remember from last year what was the meaning of this word right here? Cyan. Okay, it was a blue-green color. So this particular bacteria has a blue-green color. And because of it, we often thought it was an algae. There's a blue-green algae, but it's actually a bacteria. These bacteria like to stay together, one connected to the other, in long, skinny filaments. As it says here, they were originally, because we thought they were algae, and they're blue-green, and they make their own food, they must be a plant. Okay? Now, they don't have the other characteristics of plants, so that's the reason why we took them out of plants and made them their own kingdom. Okay? Remember, they're a bacteria, so they're prokaryotic. The blue-green color allows them to carry on photosynthesis, so they can produce their own food. So instead of living off of other organisms, they can make their own food. Photosynthetic. This one is, living as a colony. A whole group of them all connected together. But they're also capable of living independently. Unicellular. So what does unicellular mean? Uni means what? One, okay? So they can exist as individual cells. If they're in long chains like this, then they refer to the chain of bacteria as a filament. They call it a filament. In that filament, as you're looking at it under a microscope, you will find some cells that are empty. It looks like there's nothing inside these cells. I mean, there is something in it, but not the typical structures that you would expect to find inside of a cell. And so these colorless cells that are found in a filament every once in a while are called heterocysts. Heterocysts. Okay, so how do bacteria reproduce? One of the ways that they reproduce is a method that we call binary fission. Okay? They just split into two cells. It's not quite the same as mitosis that has a regular prophase, metaphase, anaphase, telophase, and a period of time in between where they're just growing. Okay, so it's not involved with mitosis. Okay, it's just a way of dividing into two. But there is a way that accomplishes the same thing as many organisms are able to do with having like a sperm and an egg, and you can get some variety that way. Well, some of them will do at certain times, they will grow a little tube that connects the insides, the cytoplasm of two bacteria cells, and they can transfer some of their genetic material between the two cells. And this is where the plasmids come into play, like I mentioned earlier. It can move some of the genetic material from one cell and put it into the other. So now this cell has not only its genetic material, but it has some genetic material from another cell. So that's going to create more variety and increase their chances of surviving in maybe an unfavorable environment. There is another thing that bacteria will do is if we have a cell over here that breaks open for let's just not worry about why, but and the genetic material like chromosomes leave this cell, and so now they're floating around in their environment. If they touch a bacteria, that bacteria could take that genetic material inside the cell and incorporate it with its own genetic material. Just, bring it in, make it part of their own. So that's kind of not, found in most living things, where you just have genetic material around and you